There's so many things you have to click. So anyway, we have two X's. All right. And in embryonic development, what happens is in every single cell, one of those X's basically is turned off. So you have determinant factors that are in the cells that just randomly turn off an X chromosome. And then the inactive X becomes a Barr body. Okay. Now, the neat thing about this is that as an adult, environmental factors can actually switch that X chromosome back on, which is kind of cool. And that's part of epigenetics. And so the genetic makeup of individuals can actually change when they're adults. And that's where we're finding, especially like in France, they're doing these twin studies, identical twin studies, because, you know, their chromosomes are exactly the same. And so what they're finding is that their genetic makeup is different from one another. And that's based off of their environmental factors, okay, that are affecting it. So that X at some point along the way could be turned back on and change actually their genetic makeup, especially if you have a heterozygote. Now, that doesn't mean that the other X is going to be inactivated, okay? But the second X, if it is like the recessive allele, could have an influence on the condition, okay, or the traits that that individual is going to express, which is kind of cool. All right, so anyway, if you're homozygous dominant, the cells, like let's say every single cell in the body, in the organism is homozygous. Well, of course you have two X's, or what am I thinking? What am I thinking? Oh, homozygous for a particular gene. Okay, one of those X's is going to be turned off. If you're homozygous, it doesn't matter. But if you're heterozygote, on the other hand, and that gene is located on the X chromosome, okay, and one of those X's is turned off, and the one X that is turned off has the recessive allele, okay, and in other cells, the dominant allele is turned off, then you have what we call is a mosaic character. And the cool thing about that is you can see that in calico cats. So calico cats are always female, okay, and the reason for that is that the black fur, orange fur is located on the X chromosome. So when you have your two X chromosomes here, this one has the allele for black, this one has the allele for orange. That one becomes inactivated. All right, this one, this cell, the black fur allele is inactivated. Now you have orange fur. So everywhere that you have cells where the X chromosome is active for the black fur, you're going to have black fur. And for if it's active for the orange, you're going to have that orange fur. Okay. And if you've seen calico cats, which you guys probably have, right? All right, you have different levels of black and orange fur. And so you might have like a calico cat. My neighbor used to argue with me that my cat wasn't calico, hers was, because her cat was almost all white and had a little bit of black and a little bit of orange. My cat looked more typical of a calico cat. It was almost even, like black and orange with a little bit of white. But when the X inactivation takes place in embryonic development, determines how much of that black and orange fur that you have. So if X inactivation occurs very early in embryonic development, then you're going to have a cat that has a lot of black, a lot of orange in it. If X inactivation happens later in embryonic development, then your cat's going to be more white with a little bit of the black and the orange. So when that X inactivation takes place in embryonic development, is also a determining factor of how much of the mosaic pattern is actually expressed. Any questions with that? So I always think that's pretty cool. And I was trying to explain this to her, but yeah, it wasn't working. So I kind of gave up because it determined that her cat was calico and mine wasn't. All right. So that is X inactivation. Like I said, new studies are showing that with epigenetics, environmental factors that influence our genetics, that some of those X chromosomes can actually, those bar bodies can actually be reversed and turned back on, which is really cool. All right, so we have linked genes. We already talked about what linked genes are. So those are genes that basically are inherited together, okay, and they're located on the same chromosome. So we have Thomas Hunt Morgan, again, working with the fruit flies. He noticed like these different patterns, patterns that were like a little bit different from what Mendel had studied. And so what he did is he took those fruit flies with body color and also with wing size. So you have like two traits that he was actually following the pattern. Okay, and so basically what he did, you have, and when you do fruit flies, so this is like, you have a black body and vestigial wings, or normal body and vestigial wings. And then the other trait would be like a black body with normal wings. And so anytime you have the plus with the fruit flies, that little plus means the wild type. And that's basically the dominant trait. Okay, so when we say that this is a hybrid, anything with the plus is the expression of that dominant trait, and then this would be the recessive trait. Okay, and so he did a test cross, and when he did the test cross, you should get like 50% and 50%, right? A 1-to-1 ratio when you do a test cross with a heterozygote and a homozygous recessive. That is what you expect. Okay, so here's what he was saying. Now this should say same chromosome. All right, and so when he tested this, the predicted ratios is if those genes were located on different chromosomes, you would have, because you're working with two different traits, you would have a 1-to-1-to-1 ratio. Okay, if they're on the same chromosome, then you would just have a 1-to-1 ratio. You would only see the phenotypes of the parents. When he did that cross, what he found, these were the results that you got. So it didn't match either one of these predicted ratios. Okay, so you had to think about this and why was that actually taking place? All right, and so he was saying that there was something physically that had to take place where you were getting those kinds of ratios. And what was that? What reason did he come up with? They were linked. What else? If they were linked, where else are we getting those recombinants? So any organism that has a different genotype from the parents. All right, so if we kind of go back, these are the parental genotypes right here. Okay, so you have your heterozygote, you have your homozygous recessive for both of the traits. All right, these two right here are different from the parental. So these are the parental genotypes. These are what we call recombinants. And anytime you have recombinant genes, you have recombinant organisms, they have a different genetic makeup from the parents. Okay, so that's a term that as biologists, we should know what that means. Okay, so there's another reason for it, and Mendel explained this when he saw differences from the parental genotypes crossing over. Okay, so those linked genes, okay, those chromosomes not only are on the same genes, but we're getting those recombinants because of the crossing over. Okay, and so we had, and I know I'm skipping around with this a lot, but I'm just explaining it as we go along. So the mechanism for having those recombinants was explained through the crossing over. Okay, so here's what we can do. We can look at what is called the recombination frequency. And the recombination frequency basically is how much of the recombinants are produced in that particular cross. And so you take the number of your recombinants, all right? Those are the organisms that have a different genotype than the parents.
So these are the parental genotypes. These are the recombinant genotypes. We add those up and divide by the total. That gives you your recombination frequency. But Thomas Hunt Morgan, one of his students, Alfred Sturtevant, he took this a step further, okay? And so what he did is he actually then looked at combinations of alleles, okay, to come up with where the locus is. And when I say locus, what do I mean? The location, okay, of those genes or of those alleles on the particular chromosome, okay? And so what he did is he would look at two of them and calculate the recombination frequency between those two, and then look at another two and look at the recombination frequency of those two different alleles, and then another two. And so now you have all these different recombination frequencies of the alleles that are on that chromosome, and you're able to actually place them in specific locations. Okay, and that is what we call gene mapping, and that is what we're going to do today. Okay, and so what Sturtevant said, and he called 1% of recombination frequency, he called that one map unit on a chromosome. So when you do gene mapping, that's what we're doing, is we're looking at the percentage, and that percentage, so if you have a 19% recombination frequency, that is 19 map units on that particular chromosome. Okay, everybody got that? All right. So, and this is an example of what he found. So this would be body color. The B is body color. CN is cyan. So that is, I think that's eye color. And then VG is wings. Okay, so these are three different alleles. And so he found that there is 17% recombination frequency between these two alleles. So that's the thing, like he would have to look at those individual alleles and calculate the recombination frequency. And then 9% and 9.5%. Now notice, if you have 9% between the B and the CN, and then 9.5 between the CN and the VG, does that add up to 17? No. Okay, so it's not exactly 17, especially if you have the half units. So it's approximate. Okay, and we're going to find that on one of the examples that we're going to do. Okay, so that is what we are going to do today. And how did they come up with these? The cytogenetic maps. When we say cytogenetic maps, you know how whenever you do banding patterns? Well, that's what a cytogenetic map is, are those banding patterns. So like each band would represent an allele, all right? And they did their studies, so they knew like what the allele was, okay? And then they can calculate the recombination frequencies based on those cytogenetic maps. All right, so that's what a cytogenetic map is. Okay, and here's another example that we have with the different characteristics of the fruit fly and the gene mapping of those mutant phenotypes. Okay, so for the PowerPoint that I'm going to go over today, what I want to do now is to look at the worksheet that I gave you. And I did not give that to Trace or Skylar. So, Allie, when you go, you want to take a worksheet for them?
Yeah, sure.
Okay, great. Okay, so, oh shoot, I thought I had that. Okay, let me pull up that worksheet. Did you guys do gene mapping at all in Bio 1?
Yeah, we did.
Did you?
In Jesse's class, we did.
Well, okay. So when you look at your worksheet, we're going to start with the side that does not have like the little data table.
so I don't have the size numbered. But here's what you want to do. You always want to start with, you want to start with the largest percentage. And that makes it a lot easier when you're doing it like that. Okay, so we see that our largest percentage is between what? A and B, okay. And so all I do is I just kind of draw a line with my two ends and I'll label this A and B. Okay, and this is between A and B is 35%. Okay, and so you kind of have to look at these. And so everything needs to add up pretty much. Okay, so if we take a look at then B and C, so C can either go 10%, 10 map, and remember each percent is a map unit. So between B and C, you have 10 map units, right? So we can either go over here for 10 map units, or we can go over here for 10 map units. And that's where you have to kind of look at everything and see how it's going to fit. Okay, so here's the thing. If I went here, if I put my 10 map units over here for C, I'm going to look down and see what else percentages I have for C. And I noticed that in letter D, I have 25% for C and A. Okay, but if I put that here, well, this is already 35%. And if I go out to here 10 more percent, then that would be 45% or 45 map units between A and C. So it doesn't make any sense to put it after C or after B. Okay, does everybody follow? So we want to put our C, okay, 10 map units in from B. All right, and so we're just going to label that and put our 10 map units. Okay, and so if we subtract that 10 map units, then between A and C, we have what? 25. All right, and we notice that between A and C, we have 25. Okay, so now, so does everybody follow what I'm doing? Okay, so now we have our A and B, we have our B and C. Oh wait, I'm on the wrong one. I was looking at the second problem and it wasn't adding up to me. Okay, so we have our A and B, we have our B and C, and we have our A and C. What's left? So what do we need to do now? We have C and D and B left, right? So C and D is 15 map units and D and B is 25 map units. So where do I need to put D? Between A and C. Okay, so we're going to put D right here between A and C. All right, and so A and D is, or let's see, between C and D, all right, is 15 map units, right? And then between D and B is 10 map units or 25 map units. And that makes sense because we have 15 map units here and 10 here, and that equals 25. Okay, does everybody follow that? So everything should add up. All right. So why don't you guys try the next one?